This method can be used for the quantitative evaluation of sarcomere maturation in iPSC-derived cardiomyocytes. Using this super-resolution-based approach, it is possible to detect even subtle alterations in sarcomere organization, which is not possible with conventional confocal imaging.
At least 3 hours before use, switch on the microscope and bring the sample to room temperature. When the microscope is ready, add 300 microliters of palm imaging buffer to a well of labeled cells and insert the chamber slide into the stage holder of the microscope. Select the 1.57 Na 100x oil objective and use the palm mode in the imaging software and activate the TURF settings. Set the number of frames to 5,000 to 10,000, the UV laser power to 0.1%, and the 647 laser to 0.2%, and the gain level to 50 to 100. When all of the acquisition parameters have been set, switch on the laser illumination and select a target cell. To acquire an image, increase the 647 laser power to 100% and reduce the gain to 0. Bleach the target cell for about 5 seconds. Then, increase the gain to 50 and initiate the palm image acquisition. For reconstruction of the palm data, at the end of the acquisition, open the data in ImageJ.